to this seminar. I'm very happy here to present a guest that quite often comes to KTH, Professor Heikki Tenhu from Helsinki University, professor in polymer technology, polymer chemistry, and we've known each other for quite a few years. It's always been fun to work with you. He will give a presentation today about functional polymeric nanomaterials, and I think I will give the word straight to you.
Thank you, Mats. Just a second. Hjälp. Can I put it over there?
Try to fix that.
No niin, tack. So, good morning, alla. It's really a pleasure to be here. I'm, as you know, I'm coming from Helsinki, as Mats said. I came here for Markus' PhD defense, which is in the afternoon. And in the afternoon, this will be very much on sustainable chemistry, which is not the case now. This morning, I give a talk about things we are doing at the moment in Helsinki. And these things, what we are doing, this is not very sustainable, because this is not the focus of our studies at all. We are mainly interested in various phenomena dealing with aqueous polymer solutions. So we synthesize and study various amphiphilic polymers, polyelectrolytes, and study the phase solution properties, and especially we are interested on so-called smart polymers with on the phase separation processes in water. So, as a stupid start, I show you a functional polymer, which is not what I'm going to talk about. Evidently, this is a polymer material which has a function, but it's not a functional polymer. And this is more like an example of what kind of polymers we are interested in and what we are dealing with. This is a cryogenic TEM image of a water droplet where we have dissolved our amphiphilic polymers. They are star-like polymers, several chains grown from one point. And in these stars, the inner block is hydrophobic and the outer part is hydrophilic. So the polymers, they form micelles in water, these dark spots here. And actually these dark spots that you see, they are the hydrophobic cores of these micelles. The aggregation number is around 200 in these. And then the hydrophilic polymer forms a shell around. And this one, it is a cationic polymer, or it is this amine, so it gets protonated in water. The particles, the micelles, they are positively charged. And this is why you see the distances between the particles are more or less constant, because they have this electrostatic repulsion between each other. So this is the world we are working with. It's not so far from real life, actually. You see, this is taken from a beach in Helsinki in summer. I don't know if Swedish people have more tendency to aggregate. In Finland, we keep distance like those charged particles. So this is what I'm indeed going to talk about. Paul Flory studied these things in 1940s very carefully. The phase transitions, solubility of polymers and solubility, insolubility of polymers. And in general, if you look at the phase diagram here, concentration versus temperature, we have in principle two possibilities. The polymer is usually soluble at under some certain conditions. Here we have a single phase region. But depending on the polymer, it may phase separate either upon cooling or upon heating. Paul Flory in 1490s, he studied polymers of that time. They were non-polar polymers in organic solvents, and he usually observed this quite logical phenomenon that when you cool solution, the polymer precipitates. Then later now, for example, we work mainly with water-soluble polymers. So they are polar polymers in polar solvent, and we usually observe this phase separation upon heating. And in this phase curve, the minimum here, it is called the lower critical solution temperature, LCST, and this one is called upper critical solution temperature, UCST. And what I plan to do today, I show some examples of this. First of these LCST polymers, actually kind of 1.5 examples of those. And then I go to, which I nowadays find much more interesting, into these UCST things. So what makes polymers undergo phase separation when you heat an aqueous solution? It is because let's take, everybody knows the polymer, and at room temperature this polymer is highly H bonded with water molecules, but when you start to heat the system, the hydrophobic interactions increase and the hydrogen bonds between water and the polymer, they break and the polymer aggregates. This aggregation process then leads to phase separation, which may be precipitation or formation of a colloidal dispersion. Whereas then these UCSD polymers, they are polymers that have very strong interactions between each other, between the chains, ionic interactions, strong H bonding, or something like that. And this is why they, for example, at room temperature do not dissolve, but they behave as what teachers say at school, that solubility increases when you increase temperature. They behave in this way, though this also, it is not like adding sugar to coffee, because the difference is here that the dissolution is a quick, step-like process, that it's insoluble at this temperature, but then suddenly it dissolves when I heat it up, when these bonds between the polymer chains break. And so I will show you now examples, and this is not from our work, but if you can see this tiny picture, the polymer I will start with is the one shown here. It's polyvinyl caprolactam. It is a polymer that behaves quite a lot similarly to polynipalm, but is different in many senses. For example, this is nowadays very much regarded as a biocompatible polymer. So it is, at least in this sense, it's interesting. And this is from this Wu's paper where they studied this phase separation process. But this shows how we assume that these polymers in general behave in water, that we heat the polymer, they aggregate, and the aggregates bind together and precipitate, phase separate. And to start with this polyvinyl caprolactam, I have worked with this polymer for a very long time. I got first time it in my hand somewhere in the 90s when a visitor from the Moscow State came to Helsinki with a polymer, self-made polyvinyl caprolactam, and we started to study its properties in water and its interactions with surfactants and so forth. And after that, eventually, we have many times come back to this quite nice polymer. And in this work, which is a PhD thesis by Jonas, I should have the draft of his thesis on my desk when I go home. So he's finalizing his PhD. Well, he mainly has made microgels and nanogels of this polymer. I will talk about them more. But we also tested, can we do PISA polymerizations with this monomer? Do you know what is PISA? Not that in Italy, no. Well, Eva knows, Mats knows, but do you know? No, okay, good. No, it's not good. Shame on you. PISA means polymerization-induced self-assembly. It means that we polymerize something under conditions that the monomer is soluble, but the polymer is not. So that when monomers start to polymerize, suddenly at some certain critical chain length, the polymer starts to aggregate, it collapses, and it builds up a particle. And we can control this particle formation with some hydrophilic additive. It can be a macro initiator, for example, or in our case, it was a macro chain transfer agent, because we polymerize this in kind of a rough polymerization. It's a little bit modified rough polymerization. They call it Madix. But in rough polymerization, you use chain transfer agents. We have a chain transfer agent which is made on polyethylene oxide, this blue chain, and this is the chain transfer unit. It is in the end. Who is doing this? It's me. So the polymerization starts, and this red one starts to polymerize here, like this. And well, then we are doing the reaction at 50 C in water, and it is about the LCST of this polymer. So the polymer is not soluble. It forms a particle. It may be vesicle, micelle, a tube. It depends on the ratios of these blocks. We don't look at those reaction equations. It's just how the chain transfer agent was made. But now here then, yes, we can do PISA polymerizations with this. But those who seriously do PISA reactions, they do polymers that polymers are not soluble in water under any conditions. This is soluble at room temperature. So after the polymerization, we have a nice milky dispersion, 50 C, but when it cools down, the polymer dissolves. But no problem. Because we have worked a long time with this polymer, we knew that we can cross-link it, for example, simply with salicylic acid, which binds to the polymer with hydrogen bonds. And so we can, in this way, we can stabilize the particles after the PISA reaction. And I finished this example with this figure. These are the SEM images, not TEM images, of course, of the particles made under different conditions. We have got here. You see, we have got clearly core shell structure, nanoparticles. But here, this has been a vesicle. There is something empty inside here, even two empty pair bases. But it seems that it was. Sorry for that. It was a. I'm getting emails. That's it. Sorry, I got this week ago. Now I'm not sure how to make it quiet. No. So there were vesicles, but when we continue the polymerization, the vesicles, they have grown inside, and so we get these blueberry-looking things as product. But these are not stabilized, and this also says that we may find some use for this kind of systems in future, because, well, now we have a polymer carrying aspirin inside. So we could use this as controlled aspirin release system. But if you take aspirin into something else, we can maybe proceed with this project. But as I said, mainly Jonas worked with cross-linked particles, cross-linked nanogels. They are usually something like 100 nanometers in size. And this is, for example, an image of these gel particles which have been decorated with tiny gold particles to 4 nanometers. they are bound here with click chemistry to the surfaces. We wanted to see, because we know that nano gold absorbs visible light and the particles heat up. We wanted to see, can we make these particles shrink with visible light? And yes, we can. This is the particle before irradiation with blue light, and this is after the irradiation. Nothing dramatic, which is understandable because these heaters are on the gel particle surface. If they were inside, the effect would probably be bigger. However, we also could make this with radio frequency electric field, make the particles shrink. And well, this may be useful for something. I don't know yet for what, however. Maybe the most useful thing we have done this with particles is that we have just recently finished a work where we bound sugars on the surfaces of these particles. It was our co-author is Philip Ekholm. He synthesized these glucose and maltose derivatives with azide functionalities. You see azide either bound directly to the ring or through a spacer, both in glucose and maltose case. And this, of course, is because these sugars, they recognize some certain proteins, and they can be used as in targeting these particles somewhere, for example, in the human body. I don't need to explain this to you, but well, maybe an important trick in our synthesis was that we made these particles in a traditional emulsion polymerization. However, we had to add there this triple bond for the click chemistry. These are very hydrophobic, and so we did this batch polymerization where we first polymerize the particle, 1 hour polymerization time, and only after that we add this hydrophobic monomer to ensure that it is on the surface of the particle. And we have seen that it works. NMR. This is maybe too much, but this is light scattering data, and it is here because this is kind of indication that our particles are very homogeneous in size, because we were managed to exceptionally well plot the light scattering data on these particles on this kind of plot where we have the inverse structure factor over against G scattering vector times radius of gyration to the second power. These are different theoretical models for different geometries, and we can see that this red one is the plain polymer nanogel particle. You see the points are here. Then we add this propargyl acrylate to the surface. It changes to here, and then we add the sugar. It changes again. And I don't go into details what are these models and what they mean, but this shows us that the gel particle, before we start to do anything to it, it's a very swollen, very soft, almost like a polymer coil. When we add propargyl acrylate to the surface, it gets compact. It's like a compact sphere. And then we add sugar, it again swells. And it's again a very soft, swollen particle. So the surface has a huge effect on the structure of this little jelly. And then what was the aim of this work? We tested, do these particles recognize the lectin protein, which is a plant extracted from plants. And this test shows we add nanogels to polymer solution, protein solution, and see how much precipitates. Pure gel doesn't precipitate anything, but when they are coated with sugars, they work. And you see that especially particles with maltose on the surface, they very effectively recognize lectin and they precipitate. So this is nice and easy. Everything works as was expected. And before I go to these UCSD things, I try to be very quick. I show another case where anything worked as we expected, which makes it, of course, very interesting, but it would take quite much time to discuss very much in detail. Fabian Puch, he finished his PhD a few months ago. He's in Germany now. He synthesized a series of diblock copolymers, which are made of polylactide and polyisopropyl oxazoline. Polyoxazolines, especially this one, they are also these responsive polymers, behaving very much like as polynipalm. So dissolve in cold water, precipitate when we heat the solution. We were expecting that this will be a very ordinary work, that the polymers form micelles with polylactide core and then a responsive water-soluble shell around. The synthesis, cationic ring opening polymerizations to make oxazolines, to make the lactides, then N-group modifications, click chemistry, we get the block copolymer. And well, what we wanted to do, we wanted to make these micelles. We dissolve the polymer in THF, which is good solvent for both blocks. We change the solvent in water. We should get particles, as I explained already. And then comes a surprise. This is not very clear. I hope you can see these dotted lines here. This is transmittance of polymer solutions. This is pipoxy. This is polyoxazoline dissolved in water. At room temperature, transmittance is 100%, so it's absolutely transparent, clear solution. This is the behavior what we were searching for. We heat the solution, vom, transmittance drops to 0. It turns milky. However, then we take our micelle. We were expecting it has a pipox shell, and we do the same experiment and we don't see any changes, which is really disturbing. Störande. We can see it with DSC as well. This is the pipox, nice endotherm when the polymer phase separates, and this is our expected micelle. Well, hardly not anything. So we had to take a step back, and Fabi started to study these polymers in bulk. And interestingly, we observed that these two polymers, they are miscible in a melt. You know that chemically different polymers usually do not mix with each other. So they want to phase separate. These are miscible in the melt. They are both crystalline polymers. And so then when we cool the system, there is a blend of two homopolymers, there's a block copolymer, then the crystallization induces the phase separation, which is of course different in these two different cases. So we saw that, all right, these two blocks may be miscible with each other, at least they are in the bulk. But they clearly are it also in these micelles, in these small particles. This is, for example, the NMR spectrum of the pipox and our particles. So all the oxazoline signals disappear because the polymer is mixed with polylactide. It's inside the particle. So we cannot observe it. So, thing which we were expecting to be extremely simple turned out to be complicated. So this is our suggestion how polymers with different lengths of these blocks, what kind of particles they build up. Because by varying the lengths of these blocks and also varying the lactide. You know, it can be D-lactide or DL-lactide, crystalline amorphous. We could make particles where everything is mixed inside the particle, or where some polymers form dangling chains on the surfaces. So there is still a lot of interesting things to study with these LCST polymers, but I try to be fast, but I want to tell you about polycations, with which we have been working for a few years now already. Polycations, as all polyelectrolytes, of course, by definition, they should be extremely soluble in water, and they are. But we observed long ago that, and of course, this should be not so surprising. By playing with the counter ion, we can change the solubilities of these. For example, this one is, it looks like an ionic liquid, doesn't it? This one, it is very well soluble when we have, for example, this chloride counter ion. If I change it into this is an anion very much used in ionic liquids. The polymer turns totally insoluble, regardless of temperature. But if I take this one, not as hydrophobic as this one, there's a triflate anion. I get a polymer which does not dissolve in water at room temperature, but upon heating it suddenly dissolves. This was fun, and we have studied this with very many systems. I skipped this. There are too many examples. But I want to tell you about this polymer, or this type of polymers. This is work by Vikram Badam, who is doing his PhD on this topic. He has synthesized a series of polycations. Polycation is this styrene derivative. You see this positive charge here. This was also made with Raft reaction. So we had a chain transfer agent again made of polyethylene oxide. It had a long alkyl chain. We get an alkyl chain substituent to this end of the chain. And so if we have here chloride, bromide, iodide as a counter ion, it's soluble polymer, but we took triflate as the counter ion. And we started to begin with something. Vikram made one polymer. We did not, at that point, we did not even think about the molecular mass effects on this, but he made one polymer, dissolved it in water. Actually, he dissolved it in an aqueous triflate solution, and it behaved in a very weird way. And actually, this photograph is important because it tells the whole story. Everything I explain after this is just, desperate trials to understand what is going on here. You see that, the polymer dissolves when we heat it. At 55 centigrades, we have a very transparent, clear solution. We start to cool it down. It turns turbid, 45 C. And then upon further cooling, it starts to clarify again. The polymer does not dissolve totally anymore, but something in this aggregation is kind of reversed. And we wanted to start to study this effect. This is the rough synthesis. We don't want to. In this table, only important thing are these red numbers, because finally Vikram made 4 polymers, varying the degree of polymerization of this cation. The polymer with which we started was this 81. He made a shorter one and 2 longer ones. And now there is a big difference, and I will now talk about the short polymers and the long polymers because they are very different. Very much data on one slide, but here are the transmission curves of these polymers with different concentrations. We see that the transmittance increases stepwise when we heat the solutions. So, and kind of phase diagram shows that yes, it is a UCST process, which you can also observe with DSC. Vikram so much likes to play with this PowerPoint. But here actually we see what we saw in those photos. Here is the transmittance curves for this same polymer of which we saw the photograph. He measured, there's again different concentrations. The salt concentration is kept constant, but we change polymer concentration, and we measure those during cooling the system. So we see what we see in the photo. Clear solution, turns turbid, and then it comes clear back. But you see that there are several steps in this process. So it's getting more interesting. We are still very confused. What is happening there? There's a phase diagram. And what is super interesting is here are the transmittance curves for block copolymer. The scale is different, but we see here this two-step transition upon cooling. If we take a homopolymer, this red curve, with a homopolycation, we see only UCST behavior, one-step dissolution or one-step phase separation upon cooling. And if we mix the polycation and PEG, we see only this same process. And this is fun. To see this effect, we have to bind these two polymers covalently to each other. They don't attack each other if they are separately dissolved in water. But this is because of the Bock structure of the polymer. And that is cute. And then it turns out that indeed they are these two short polycations which have two transitions, and then the two long polymers which only show UCST. And here is a combination of these. Two short polymers and two long polymers. Very different. Well, but this already hints that the PEG is a PEG 5000. We keep that constant. Well, this already tells us that there is something between PEG and the polycation. When the polycation gets very long, there is kind of free polycation which does what it wants regardless of PEG. And this is also fancy because you saw that because of the rough polymerization, we had in the polymer a very long alkyl chain. And many people have studied this kind of effect of these hydrophobic tags on polymers, but they have done these studies with non-charged polymers. If you take, for example, this polynipalm and add this alkyl chain to the end, it most probably in water, it forms a flower-like micelles. These polyelectrolytes do not do that because they are so highly soluble. But we had to check the effect of the end group, so we removed it. And these two sets of curves here, they are from the shortest block copolymer measured in two different salt concentrations. Salt is very important in this process. 100 millimoles of triflate, 200 millimoles of triflate, very different process. But The dotted lines are the polymers from which, these are the original polymers, and the solid line is the one from which we removed the end group. So in this case, the end group hardly has any effect. And that was good to know, of course. Back to the photo, you can see the same thing with light scattering. It's just normal, more light scattering. But I'm coming to the end. But this is super interesting, I can tell. NMR of this, these polymers in a triflate solution at different temperatures. What is so special here? This is the signal coming from polyethylene glycol, polyethylene oxide. And when we have a closer look, we see that it divides in two. This I think I have never seen before. Here there are the PEG signals enlarged. This is the longest polymer. This is the shortest. We always see these two peaks from PEG. And because of this chain length dependence, in a short polycation, this one is quite weak. In the long polycation, it's strong. So it maybe tells us that this comes from the free PEG and this from the PEG, which is closely interacting with the polycation. Either oxygens and the cations, they of course bind to each other. And salt is important. If you repeat this measurement in pure water, just one signal. Now this tells that the triflate ion, it mediates this interaction between the cation and the ether oxygen. So without the triflate ion, we don't see these fancy things. So after all this playing, I made this story now short. Maybe I should not have skipped, for example, the light scattering measurement data. But this is now where we have ended up. We are now suggesting that in the case of a short polymer, when we cool the transparent solution, the polymer phase separates, we get this sort of particles where the polycation with its counterions is here in the core, and PEG chains are shielding it. Whereas then when the polycation length increases, this is the longest polymer here, more or less everything is packed again in the same particle. These are particles which have cationic surface potential, zeta potential. These do not. So we see that we saw that this PEG polycation interaction, which is mediated via the triflates, is the thing that makes this phase separation so complicated process. This is where I think I will now end. If you now, please don't ask yet what are the applications in mind. I have no idea, because at the moment we are just studying these phenomena. We have been busy enough trying to understand what is going on here. But honestly, why we quite a while ago started to make polymeric ionic liquids, which you could also call this in that way, is that even in my chemistry department, in organic chemistry, they synthesize ionic liquids, and you know or don't know, they have, together with Aalto University, they have developed a process where they dissolve cellulose in ionic liquids. Then they make fiber of that. A world's most expensive way of making Marimekko dresses. But of course, we know that, and it's been known for ages, that polymeric ionic liquids, they dissolve almost anything. And of course, I continuously have had the idea that we, at some instant, when we understand everything about this, we also want to see what these particles can eat from aqueous solutions and whatnot. So, thank you for your listening. You didn't interrupt me at all, but maybe you have some questions or comments. Well, it never happens in Helsinki that somebody interacts.
Thank you for a really great presentation. It's
Thank
super
you.
nice to see how you can master the formation of these structures by varying the architecture. So I'm a little bit curious about this microcalorimetric technique that you are using.
Mm-hmm.
So are you studying also the phase transitions in the micro-CT, or do you do it mainly by transmittance?
Micro-CT?
Yeah, microcalorimetric. Sorry.
Yeah.
Sorry.
Yes. Sorry, I did not really pay very much attention to that. But for example,
the main workhorse in this work is the transmittance.
Actually, this most informative, the simplest and most informative way so far. But this, indeed, this is the heating curves in microcal. Of course, in these cooling curves, you can also see that there are steps in this process. Of course, we have tried to get a closer image of this, but well, we get kind of similar information from this.
But when you use the microcalorimetric, what sample sizes can you work with? I'm curious how if it would be possible to study, for instance, the absorption of such particle on nanocellulose, for instance. But I guess that we would need quite a large sample volume to get good measurements.
Oh, they are small. I, of course, do not remember. I don't remember when I previously went to the lab, actually, but it's less than 1 milliliter.
Okay, but it is that much.
And of course, when you talk about your cellulose, I get worried because the sample, it's you have to inject your sample from here to the cell, meaning that you have to be able to get it out as well. But so far we have always got our samples out. I would be very interested to try, of course. Why not? Binding or what?
Yeah, any kind of those particles, if we have negatively charged CNS.
Yes. Then I'm not sure what heat effects are associated with this process. Yeah, worth trying, certainly. Yeah.
So you have these two steps here. Do you have any proposal what it is?
Yes. So this is now a short polymer, and I cool it. It starts to build up this particle, and it is somehow some sort of reorganization in this particle. You see that I don't know yet, because at this instant we have the particle which is shielded with PEG, but of course, it's not in the beginning. This is kind of formation of it. Here we have huge aggregates. Where was the light scattering that I skipped there? There is a reason why I skipped this. But 55 centigrades, we see this is an artifact. I have to say something about this. But at 55 C, we see if the polymer is dissolved, we see something about 10 nanometers in size. This one peak. We cool it to 46, 45, this temperature, very big aggregates build up, and then at 25 it is these reorganize. They don't aggregate further, but these particles somehow reorganize. These are the two steps. It's fun that when we sent this to macromolecules, I said that because 15 years ago, 20 years ago, when light scattering technique was new, everybody wanted to do light scattering, and everybody pretended they know everything about light scattering theory. And so I'm from that generation. So I said that this is just a polyelectrolyte effect and we don't need to care of that. So the referee asked me, what is a polyelectrolyte effect? Luckily, Chi Wu was our editor. He knows light scattering, he knows everything. So I just said that this is the effect often observed with charged polymers in low salt concentration, and put a zillion references there, and it was good. Yeah, but people nowadays, they don't anymore care about stupid theories behind this kind of technique because they are automatic. You just push the button. But I hope I answered your question.
But do you think you're sure you have spherical shapes all the time?
Yes. As far as we know at the moment, yes. We have not done proper analysis on that, though, I have to admit. And it's good that you say it, because this is what we need to do. This is what we are doing, or what Vikram is doing in Helsinki at the moment. It's Friday, he should be working. Yes, we try to verify this with fluorescence measurements, so that we take anionic fluorescence probe. We also use, of course, pyrene because it's standard, but we take pyrene sulfonate because we can expect it sitting here in the core. We can observe its fluorescence, and then we add a quencher to the water around. Now there should be, if this is correct, there should be a difference in the rate how the quencher diffuses into the core. But it's now still under construction. I put on the list and the analysis of the shape. Yeah.
When you added the proprietary acrylate to at the last stage,
Yeah.
it sticks to the surface. But you also said it is very hydrophobic. How come it does not migrate inside or swell the particle?
That is a good question. It seems to work, and we see it in that an indirect way, so that this is now a thermosensitive polymer, and of course we studied this lectin binding at room temperature and at elevated temperature, and there was no difference. People have done similar experiments with polynipalm particles, but they always mix all the monomers right in the beginning of the polymerization. Then the particle acrylate is dispersed through the particle. And then, of course, they see that when they heat the system, particle collapses and it can no more bind lectin. In our case, they are on the surface because the lectin binding is not at all affected by temperature. So seeing is kind of believing.
Yeah, sure.
You others may also ask. Okay. No.
So you mainly work with diblock polymers. So how would the influence be if you go for other architecture, for example, 3-block copolymers?
Are you talking about this cation?
Yeah, exactly. Or just how would that influence the behavior of your polymers?
How could I tell? I haven't done that yet. But
you would have the same problem.
This is very much what we aim to do. Yes, we've been talking about making stars of these. And even mic to arms. You know, mic to arms would be such that we would have a star where we have long polycation chains and then short peg chains. We could repeat this. Yeah, we want to play with this architecture. That's true. It's a very fantastic idea, actually. Are we ready?
Yeah. Okay, I think we thank Heikki again for a very nice time.
Thank you for coming to listen. It was a pleasure. No need.